Okay.
Okay.
Okay, you start, Ashraf.
Okay. Assalamualaikum and good morning to panel of lecturers and fellow students. My name is Noraisha binti Khairul Anwar under the supervision of Dr. Khairul Aswar. Today, I will be presenting my research, the development and user acceptance survey of Chlorella vulgaris infused strawberry honey jam. I would like to start with an introduction of my study. About Chlorella vulgaris, it is a eukaryotic green microalga capable of producing energy. It has 4 essential properties, which are high nutrient contents, high value chlorophyll, fiber cell wall, and growth factor. It promotes some beneficial biological activities such as anti-tumor, antimicrobial, antioxidant, and anti-inflammatory. Due to that, it has been used for various applications in the industry. The problem statement of this study: Chlorella is high in nutrient content and have the potential to enhance the nutrition and function of food. However, its seafood taste and smell are quite noticeable. To solve this issue, the Chlorella will be infused with a fruit jam and a sweetener. The significance of the study is to extend and explore a new innovation of food product. Besides, this study may act as a reference for the development of chlorella-based confectionery products in the future. This study aims to optimize the Chlorella vulgaris, strawberry jam, and honey ratio via box Benken design, and to conduct a user acceptance survey of the Chlorella strawberry honey jam comprising of appearance, texture, taste, and aroma. Next, the methodology of the study. In designing the experiment, there are 3 factors. Chlorella, honey, and strawberry jam were assigned to box Benken codes and given its values and units. -1 is the level for the lowest amount, 0 is the moderate level, and +1 is the highest level. By alternating this level of every factors, 15 new combination of factors were formed. To prepare the jam, a homogeneous solution of chlorella was prepared. Then, strawberry jam was added, followed by addition of honey as an extra sweetener. Finally, 15 jam samples were produced. 62 participants were tested for the newly developed jam to evaluate the appearance, texture, taste, and aroma parameters. The survey was conducted via Google Form. The statistical analysis used are box Benken experimental design, response surface analysis, and response optimization. Moving on to results and discussion. In the box Benken design, 15 jam samples were formed and tested. The average scores for appearance, texture, taste, and aroma parameters were calculated and then were analyzed using response surface analysis. In response surface analysis, in terms of appearance, for interaction between honey and strawberry jam, yielded a high response when honey at -1 and strawberry jam at +1. For interaction between chlorella and strawberry jam, yielded a high response when chlorella at -1 and strawberry jam at +1. For interaction between chlorella and honey, yielded a high response when chlorella and honey at -1. It is due to the participant preferred the green color of chlorella and the light yellow color of honey at the least amount. However, the interaction between the factors obtained are not significant. Next, regarding the texture, the interaction between honey and strawberry jam obtained a high preference when honey at -1 and strawberry jam at +1. For interaction between chlorella and strawberry jam, obtained a high preference when chlorella at -1 and strawberry jam at +1. For interaction between chlorella and honey, obtained a high preference when chlorella and honey at -1. It is due to The addition of chlorella and honey, if it's too much, will affect the texture of the jam. However, the interaction between the factors are not significant. In terms of taste, the interaction between honey and strawberry jam yielded a high response when honey at -1 and strawberry jam at +1. For interaction between chlorella and strawberry jam, obtained a high response when chlorella at -1 and strawberry jam at +1. For interaction between chlorella and honey, obtained a high response when both chlorella and honey at -1. It is due to the participant prefer the seafood taste of chlorella and the strong taste of honey at the least amount. However, the interaction between the factors obtained are not significant. Next, concerning the aroma, the interaction between honey and strawberry jam obtained a high preference among the participants when honey at -1 and strawberry jam at +1. For interaction between chlorella and strawberry jam, obtained a high preference when chlorella at -1 and strawberry jam at +1. For interaction between chlorella and honey, obtained a high preference when both of chlorella and honey at -1, as chlorella and honey possess a strong smell. However, the interaction between the factors are not significant. A response optimization was conducted to obtain the optimum formulation of the jam. With a maximal response, the level of chlorella is at -1, honey at -1, and strawberry jam at 0.33. With a consumer acceptance of 60.2%, the optimal values are 0.5 ml chlorella, 3 gram honey, and 16.5 gram strawberry jam. Lastly, conclusion and recommendations. It can be concluded that the respondents prefer the addition of Chlorella vulgaris in the jam at 0.5 ml. However, due to inadequate number of responses, the interaction between the factors are weak. Hence, it needs to increase the number of participants in the acceptance survey and create a new formulation of the jam to segregate the level of each factor. From this study, it provides an insight to develop a profitable chlorella-based confectionery product in the future. Here is the list of references used in the slide. And that's all from me. Thank you.
Okay, thank you. Aisha. Okay, thank you, Aisha. So I open for the Q&A question from the panels. I start. So I start with the first question. Okay, can you go to your results and discussion slide in any one of your box and anyone? Okay, no, the next one.
Okay.
Okay, so I just want you to elaborate what is your meaning with the interaction, with the statement interaction between factors obtained not significant. Then can you elaborate with this statement? Dengar tak?
Yes.
Okay. Yeah. You understand? So apa maksudnya bila you kata interaction tu tak significant? So what can you elaborate?
Okay. So when the average scores were analyzed in the response surface analysis, the interaction, for example, between the factors of honey and strawberry jam, obtained P is more than 0.05, as the confidence level of the graph is 95%, and the value that is obtained is not in the 95% of the confidence level, and it obtained at more than 0.05. So the results obtained are not significant.
So means that the interaction, let's say between honey and strawberry kan, so interaction dia tak bagus lah. Kalau
Yes.
daripada layman, betul.
Yes.
So semuanya, so based on this result means that all the three interaction here in this slide is not good interaction.
Yes, because not enough of number of respondents.
Okay, not enough number of students. Okay, never mind. Any question for other panels? Thank you.
Kita lihat pada if you see your number of respondents, this is 62, right?
Yes.
I think it's enough for the minimum number of samples is 30. I think it's enough, the number of 62. So all of your result is not significant.
Yes.
So it is mean there is no difference between this, your testing, right? So
Yes.
can you refer on your first objective? Okay, so just briefly explain what the result you achieve from the first objective.
Yes, pardon?
What you have, what is the result from your first objective?
On the first objective,
Yeah.
based on my study, from the results I get, then
Your objective is to optimize the
Yes.
Chlorella, strawberry jam, and honey ratio. So, what is the result from your first objective?
The result of the first objective, I was not able to optimize the Chlorella vulgaris and strawberry jam and honey ratio. Hence, I use a response optimization to obtain the optimum ratio of Chlorella vulgaris strawberry jam and honey ratio.
So meaning there's no result obtained for the first objective ya Aisyah? First objective tak dapat optimize ke dapat?
Yes.
No results for the first objective. You can't achieve the first objective.
Yes.
Okay. Itulah. Itu maksudnya kalau dia tak dapat optimize lah maksudnya kalau.
Ya, betul sebab semua tak signifikan. Bila tak signifikan, so you tak ada comparison lah. You tak boleh nak compare kan. Tapi tak apa, elongate je. Kenapa awak tak dapat nak identify, nak optimize mana satu.
Yes, correct. That's what we mean. You have to elaborate your finding tu lah, your result tu, you have to elaborate. Right? You tak boleh letak just is not significant. Maksud dia. So at least you elaborate it.
Okay.
Okay, any question? Berapa minit dah? Okay.
Lebih dah. Tadi pada masa soalan Dr. Wan, actually dah habis 10 minit.
Oh, okay. Alright, in that case, so thank you Aisyah.
Okay,
alright.
thank you.
Alright. So we go to the second presenter. Okay. Muhammad Amiruddin. Betul tak? Amiruddin?
Ya, saya.
Okay, are you ready?
Pakcik tak boleh dekat. Saya nak masukkan lagi satu.
Ready lagi?
Tak, sebab saya nak buka
Okay, saya dah masukkan Amiruddin. Lagi satu.
Sebab saya nak masuk Google Meet dekat laptop saya tak boleh pakai. Dia punya kamera dengan mic rosak.
Okay. So, boleh present ke?
Boleh.
Dah boleh dah sekarang?
Tapi saya cakap dekat sini.
Tapi dengar clear je you cakap Amiruddin.
Sebab saya yang kamera ni saya pakai dekat tab.
Dekat tab. So, you present dekat telefon?
Saya present dekat laptop.
Dekat laptop. Okay, nampak tu? Okay je.
Okay.
Okay, once you're ready, so you can start your presentation. Okay, Amiruddin?
Okay.
Okay?
Okay.
Alright.
Assalamualaikum and a very good morning to all examiners. My name is Muhammad Amiruddin Mohammed, under the supervision of Dr. Fakaruzan Raji Yahya. Today, I will be presenting my final year project entitled In-Silico Identification of Transmembrane Proteins of Salmonella typhimurium. Let's start with the background of the study. In-Silico is defined as the usage of silicon in computer chips in reference to biological experiments. Salmonella typhimurium is a gram-negative pathogenic bacteria that is typically found in the intestinal lumen. Transmembrane protein is a kind of integral membrane protein that surrounds the cell membrane. It functions as gateway, allows specific substances to be transported across the membrane. The bacterial outer membrane proteins can present attractive targets for better antimicrobial agents and vaccine development, since they are surface exposed. Computational identification using programming tools such as BLASTP and TMHMM have been introduced to accurately identify bacterial transmembrane proteins. The problem statement of this project is multiple lines of experimental works demonstrated the important role of transmembrane protein in Salmonella typhimurium. However, the study of transmembrane proteins used in silico strategy is still limited. There are still many Salmonella typhimurium proteins that remain not well characterized. The significance of the study is it can provide knowledge and understanding in Salmonella typhimurium transmembrane proteins, which may assist in the drug and vaccine development. The objective of the study is to identify transmembrane proteins of Salmonella typhimurium using BLASTP, TMHMN, and Cell2Go. The methodology of this study consists of material and methods used. For material, the radiata used is 150 uncharacterized proteins in FASTA format from Uniprot database. And for the methods, after raw data were collected, they were analyzed using 3 different websites to identify different characteristics. The uncharacterized protein sequences will be used as queries in BLASTP for identification of homologous proteins. The parameters of search against non-redundant databases will be set to e-value smaller than 1 to the power of 10 of -6 and sequence identity bigger than 30%. TMHMM is able to predict transmembrane domain and the full topology within or outside regions of the membrane. Hello uses its most recently trained model to predict subcellularization for the query protein. The Cell2Go findings were displayed as Google-created pie charts at the output interface for each query sequence, and the results were presented in tables and pie charts for better understanding. Figure 2 shows the classification of uncharacterized proteins based on biological pathways. From the pie chart, each response and pathogenesis to identify the major biological pathways in Salmonella typhimurium. Many proteins annotated involved in stress response biological pathways such as serine, threonine, phosphatase. This protein plays a role in establishing and maintaining infection and also important for the response to heat in host. Threonine tRNA is a type of protein that has the ability to withstand antibiotics. Resistance is frequently caused by mutation that prevents antibiotics from binding to the protein or by amplification of the gene that codes for the protein. Some proteins annotated that have response to antibiotics are RNA methyltransferase and type 2 toxin system HITA family toxin. Pathogenesis is the second highest biological pathway involved in Salmonella typhimurium. One of the examples is non-fimbrial adhesin SIIE. According to Elias et al., the role of virulent factor SIIE is the adhesion to epithelial cells by attachment to intestinal epithelium, and it also helps in invasion. Alpha-hemolysin is a pore-forming toxin that lyses red blood cells, white blood cells, and platelets by creating pores on the cytoplasmic membrane. Figure 3 is the second depicts the classification of uncharacterized proteins based on molecular functions. From the figure, DNA binding proteins represent the most abundant proteins among the 150 protein sequences. This result is in agreement with et al., showing that 39% among the 100 essential hypothetical proteins consist of binding proteins. The binding proteins were divided into several categories. One of them is nucleotide binding proteins. RNA binding, which contributes 10.7% of the whole molecular function, is also included in this protein. One of the DNA binding proteins is annotated as toprim domain containing protein, which introduces the replication and transcription process of DNA. Its function is to release the supercoiling and torsional tension of DNA by temporarily cleaving and rejoining one strand of the DNA duplex. CVS ribosomal protein L1 is one of the RNA binding proteins annotated. This protein is one of the primary rRNA binding proteins that binds closely to the 3' end of the 23S rRNA. The second highest percentage of abundance of proteins in Salmonella typhimurium is transperases. it accounts for 10.7% among 150 protein sequences. This result is supported by which stated that 18% of total EHPs belong to transferases. Some examples of proteins that have been annotated under transferases are RNA methyltransferases and glycosyltransferase. The function of RNA methyltransferase is to methylate cytosine at position 1407 in cysteine as RNA, and the function of glycosyltransferase is to start the initiation and elongation of glycan chains of cysteine by transferring activated sugar residues to the proper acceptor. Pyruvate kinase functions to catalyze the conversion of phosphoenolpyruvate and ADP to pyruvate and plays a role in glycolysis. Figure 4 shows the classification of uncharacterized proteins based on subcellular localization. It is found that 61.3% out of 150 protein sequences were located in cytoplasm. This result is in agreement with Saha et al. And Yahya et al. Stated that 59 EHPs were predicted as cytoplasmic and 76% of CTOF identified host proteins were located in cytoplasm respectively. The highest amount of protein subcellular localization is at enamel of Salmonella typhimurium, which is 29.33%. This result is supported by Saha et al., showing that 23 EHPs were predicted as membrane-bound. Figure 5 depicts the uncharacterized proteins containing the transmembrane domains. From the pie chart, it was found that 15.3% of the uncharacterized protein sequence were predicted to contain the transmembrane domains. This result contradicts the Freeman Jr. et al., which stated that only 2.0% are predicted to encode transmembrane beta barrels. This is because the difference in the amount of protein sequences data collected was too big. 150 protein sequences in the study. The study conducted by Freeman Jr. used 4,425 protein coding genes. The amount of uncharacterized proteins containing transmembrane domains in the study are 23 out of 150, whereas the amount of TMBBs are nearly 100 genes out of 4,425 genes. Table 1 shows the list of transmembrane proteins found in Salmonella typhimurium. Transmembrane proteins associated with pathogenesis were identified. The protein also involved in DNA binding located in the membrane part of the bacteria. In conclusion, prediction of transmembrane helices of Salmonella typhimurium is achieved by using Juniprot KBS database, BLASTP, TMHMN, and Saratoga database website. The major biological pathways predicted, stress response and pathogenesis.
Dah habis Maridin? Tak apa, sambung habiskan sikit ni.
Okay. Major molecular functions found are binding proteins, transferases activity. The subcellular localization of the uncharacterized protein is in cytoplasm. Among 150 sequences, only 20 sequences contain transmembrane domain. Transmembrane proteins are located in the inner membrane of Salmonella typhimurium, mostly associated with pathogenesis and functioning in DNA binding. This project is recommended to be further studied because the protein sequences in the Uniprot database are not well reviewed and analyzed completely. The function written is too general and need to do more experiments to show the evidence at protein level. That's all for me. Thank you.
Okay, so we open for the Q&A. Any question to the Amir?
Amir, anybody wants to ask?
Okay, silakan proceed.
Can you please go back to the study by Freeman, I'm not mistaken. Okay. Right, 15.3, because you said that this result contradicts with Freeman Jr., which stated that only 2.2% are predicted to encode transmembrane beta barrels. Okay, can you explain further? Because why do you get so different, the results? In your own words.
Okay. For this study, in this study, I only found 23 out of 150 that contain transmembrane proteins. When I was trying to find the related data, the related paper for this, the nearest result that I found is the Freeman Jr. Because there's not much paper about transmembrane domain identification in silico ever been released. So the reason why the percentage of treatment result is too small because it uses the protein encoding gene, while I'm using the protein sequences.
Okay, I see your point. So there are not many studies done on this type of proteins transmembrane produced by Salmonella.
In-Silico strategy using computational simulation is still limited.
Okay, thank you.
Amiruddin, what is the significance of proteins that have transmembrane domains? Why is it people are looking at these proteins?
Because bacteria with transmembrane domain, they are located in between the outer membrane and inner membrane, so they are more protected, and they are more protected from and vaccine. So by this study, we can identify what kind of proteins that we found or we predicted, and use the data already that have been found to develop newer vaccines and also drugs to prevent their infections in humans.
Okay. Alright. Okay. So I think time also limited. So we have to say thank you to Amiruddin. Okay. We move to the next speaker. Next presenter. Sorry. Next presenter is siapa ni? Alia binti Ismail. Betul?
Yes, ada.
Okay. So you can share your presentation slide, and you can start once you are ready. So while waiting, Syafika ada lagi tak kat sini dalam ni? Syafika?
Ada. Nama dia ada tu.
Nama dia ada. So dia dah hantar recorded belum? Kan tak ada lagi kan?
Dia hantar recorded dalam group nanti.
Itulah. Dia kata, saya tak tahu kat mana dia nak hantar recorded ni. Syafika, you boleh respon tak?
Mungkin line dia.
Itulah sebab tadi dia kata nak hantar dekat Telegram.
Eloknya hantar kat group kalau boleh kita dengar so that we can have Q&A session.
Betul. So better you hantar dekat WhatsApp ni, dekat group WhatsApp ni. Syafika ya? Alright, Alia okey dah boleh present?
Doktor, saya ada masalah untuk share slide saya.
Okay. So macam mana tu? Kita nak presenter dulu ke macam mana?
Boleh.
Kita tak settle di panel. Kita nak presenter dulu ke? Ada masalah.
Dia langsung tak boleh present ni. Boleh je, teruskan je lah.
You tak boleh present lah. Tak boleh present. Tak boleh share.
Boleh.
Boleh ke tak?
Boleh.
Dah boleh dah? Dah boleh present dah? Okay. Alright.
Okay.
Alright. Okay, so you can start ya. Once you ready.
Assalamualaikum warahmatullahi wabarakatuh, and good morning to lecturers and my friends. Today I'll be presenting about my study with the title The Effect of Colchicine on Capsicum frutescens. I'll start with the introduction of my study. As a few countries such as India, Malaysia, and Mexico have been cultivating Capsicum frutescens for years, becoming a part of their income and increasing the working rate. The high demand of Capsicum frutescens keep on increasing because of the expanding growth of the food industry, pharmaceutical, and cosmetic, according to Amana et al. This fruit that comes from the mutated Capsicum frutescens plant is considered very important because fruit which is bigger in size and weight is preferred more compared to the normal fruit that come from the normal crops, according to the Hull. Colchicine also is a very common mutation agent to induce mutation in plant. Significance of the study is it will be very advantageous to clearly see the degree of mutation that is caused by colchicine on the Capsicum fructans plant. The problem statement is that the amount of study on the effect of colchicine on Capsicum fructans is not sufficient to conclude the exact effect of the colchicine on the morphology of Capsicum fructans plant. When I say the morphology, I refer to the length of fruit, height of the plant, length and axial width of the leaf. The objective of the study is to assess the morphology of Capsicum fructans plant after the induction of colchicine at different concentration for 90 minutes and 180 minutes. The second objective of the study is to determine the suitable colchicine treatment for Capsicum fructans after the induction of colchicine at different concentration for 90 minutes and 180. My methodology consists of the preparation of Capsicum fructans seeds, preparation of colchicine solution, seed germination, data analysis, result and discussion. The first parameter that I look into this study is the germination of the seed. For 90 minutes of treatment, the highest germination rate was obtained for the control treatment itself with 10.31 per day, while the lowest germination was obtained from the 0.03% of colchicine treatment. For the 180 minutes of treatment, the highest germination rate was obtained from the treatment 0.02%, while the lowest germination rate was obtained from the treatment 0.04% of colchicine. The second parameter that I look into is the survival percentage of the seed. For 90 minutes of treatment, the highest survival percentage was obtained from the treatment 0.04% of colchicine, while the lowest was obtained from the 0.03% of colchicine. Next, for 180 minutes of treatment, the survival percentage was obtained from the treatment 0.01% of colchicine, while the lowest survival percentage was obtained from the treatment of 0.04% of colchicine. The third parameter that I look into is the length of the root, which is very important in this study. For 90 minutes of treatment, the highest root length was obtained from 0.03% of colchicine, while the lowest root length was obtained for the control treatment itself with 3.56 cm. However, there was a significant difference between the groups that were treated with different concentration of colchicine. For 180 minutes of treatment, the highest root length was obtained from 0.03%, while the lowest root length was obtained from the treatment 0.01% of colchicine. There was also a significant difference between the groups that were treated with different concentration of colchicine in this study. The fourth parameter that I look into is the height of the plant. For 90 minutes of treatment, the highest mean height of plant was obtained for the 0.04% of colchicine with 5.89 cm, while the lowest reading for mean height of plant was obtained from the 0.01% of colchicine with only 4.32 cm. There was also a significant difference between the groups that were treated with different concentration of colchicine. For 180 minutes of treatment, the highest mean height of plant was obtained from the treatment 0.04%, while the lowest one was obtained from the control treatment itself with only 4.35 cm. For this treatment, there were also significant difference detected by the ANOVA test. The last parameter that I look into is the length and width of the leaf. For the highest length and width of the leaf were obtained from the treatment 0.04% of colchicine with 3.16 cm and 1 cm respectively. However, the lowest reading was obtained for the control treatment with only 1.8 cm and 0.73 cm respectively. There were also a significant difference detected by ANOVA test. For 100 minutes treatment, the highest mean length and width were obtained from the 0.04% of colchicine with 3.28 cm and 1.23 cm respectively, compared to the lowest reading that were obtained from the control treatment tube with 2.42 cm and 0.8 cm respectively. The discussion is study. As we know, colchicine plays a role in plant growth as it produces a better root system and more robust polyploidy plant compared to typical diploid plant, according to Kakini and Rose 2010. The size and thickness of the leaf of polyploidy plant are bigger than the diploid plant, according to Allen et al. 2007. In conclusion, the most suitable and effective method in applying colchicine is with the treatment 0.04% of colchicine with time exposure of 90 minutes to induce mutation in the plant called Capsicum frutescens. When the concentration of colchicine increases, the larger the size of the leaf, the higher the plant height, and also the longer the root of the plant. Recommendation for this study is that further study needs to be done to verify the polybody level of the plant genome, such as the preparation sample of the site to observe the actual number of mutated chromosomes in the cell. Apart from that, further study is also need to be done to study the effect of colchicine on the fruit size of the plant. This is a difference that I saw in this presentation. Thank you for listening to my presentation. I open the Q&A session.
Okay, thank you, Alia. So I open for Q&A for the panels. Any question on the panels? Okay, if not, I might ask. So for your conclusion, okay, can you refer to the slide of your conclusion? So for the conclusion, you state that the best concentration is 0.04%, right? So I can see that you have test for several parameters. Yes. Let's say width of the leaf and so on. So this 0.04% is you refer to which parameters? Means that what is refers to which one?
Parameter I refer is all parameters for all 90 minutes treatment. As you can see for the length, the highest comes from 0.04% for 90 minutes of treatment. Same goes to the height of the plant.
Oh, for all the parameters, you found that this is the best concentration.
Yeah, but apart from the length of the root, because the length of the root, the highest reading was obtained for the 0.03 parameters.
But for these types of plants, so which is the parameters that normally we are is quite important for us to to know in order to find their the best characteristic for this plant.
The most important parameters are the length of root and the size of the leaf.
Size of the leaf.
Yes.
So this 0.04 is achieve your objective.
Yes.
Okay.
I just want to ask, when you talk about what you have done with your treatments, it resulted in a bigger size plant, right?
Yes.
So does that actually resulted in more fruits or how?
When it comes to fruit, further studies need to be done. Hence, I'm unable to say the effect of colchicine on the fruit size.
Okay, thank you. Time's up.
Time's up. Okay. Thank you. Alright, thank you, Alia. So thank you for it. So then we move to the next presenter, Abdul Rahman. Am I right? Bukan. Sorry. Akila Basita. Sorry. Akila Basita binti Abdul Rahman. Akila
silap nama.
Itulah sebab dia. Sorry Akila. Dia punya presenting tu nampak kat bawah je. Printing dah. Akila, dah ready? Ada. Okay, so you can share your slide. Okay. Mata pun tak nampak.
Boleh nampak ke slide show?
Okay, boleh?
Boleh.
Assalamualaikum and good morning to all the panels. I am Atila Basita, and today I would like to present my thesis, which titled as Genotoxicity and Cytotoxicity Study of E-cigarettes using Vigna radiata Mung bean as plant bioassay. The usage number of e-cigarette is rising greatly. The e-liquid flavoring entices more people, especially the young adults, to start using the e-cigarettes. However, there are many flavoring ingredients used in the e-liquid that have not been proven to be safe when inhaled. Therefore, this research aimed to study the genotoxicity and cytotoxicity of the flavored and flavorless e-cigarette liquid. So, in this study, alien test was used because the chromosome structure and functions are similar in plants and other eukaryotes. In addition, the cytotoxicity effects in plants and animal cell lines are identical and comparable. In this study, mung bean was used as a replacement for allium cepa because it sprouts in a short amount of time and it grows easily in warm temperature. So moving on to the objectives of the study. The objectives were to measure the growth rate and to determine the mitotic index and the percentage of chromosomal aberrations of mung beans after exposure to different concentration of flavored and flavorless e-cigarette liquids at 24 and 48 hours. So the experiment was started with the preparation of e-liquids of 2 different flavors from the same brand, and then it was followed by the exposure of mung beans to e-cigarette liquids with the concentration of 0, 10, 15, and 25%. The third step was the determination and measurement of the survival percentage, germination rate, germination percentage, plant height, plant growth rate, and also the root growth rate. The mitotic index and the percentage of chromosomal aberration was also determined. All the data collected were then analyzed using the two-way ANOVA and Tukey test. Moving on to the results and discussion. So here is the effect of the e-liquid on the mung bean seed survival percentage. So we can see that there is a sharp decrease in survival percentage in both flavors and treatment hours, with an increase in the concentration of the flavored e-cigarette when compared to the control. Similarly, in the seed germination percentage, both flavors of e-liquids reduce the germination percentage of mung bean in both treatments as the concentration increased at 24 and 48 hours. The mung bean seed germination rate recorded a similar result as the previous parameters tested, where a negative trend can be seen in both flavor treatments when the concentration is increased accordingly to 24 and 38 hours. So the negative trend in all of the observed parameters are caused by the presence of mutagenic elements in the liquids, which attributed to the cellular disruptions at the physiological level, which then causing a delay in the start of mitosis and chromosomal abnormalities. So on the next parameters that I'm going to explain, overall, both flavors of e-liquids reduce the plant growth rate, plant height, root growth rate, the mitotic index, and increase the chromosomal aberrations of the mung bean in comparison with the control, regardless of the time exposure. So here is so let's move on to the effect on each parameters. So here is the effect on the mung bean plant growth rate. So in 24 hours, the significant decrease in mean plant growth rate can only be seen in the flavored e-liquid when the concentration is increased from 10% to 15%. Meanwhile, in 48 hours, the plant growth rate of mung bean exposed to flavorless treatment was only significantly reduced to 0.223 cm at concentration of 15%. So the reduction of the plant growth rate is related to the decreasing of the mitotic index, which is a critical element in plant development and innovation. Furthermore, the presence of the C metaphase may also terminate the metaphase cell cycle, which then impedes the regular cell division process. The next one is the effect on the mung bean plant height. So we can see that in 24 hours, further increase in concentration to 15% shows a significant reduction. And on the other hand, in 48 hours, flavored treatment shows a decline in plant height when the concentration is increased to 25%. In contrast, flavorless e-liquid reduce the plant height at concentration of 15%. So So the Mung bean plant height reduction can be linked with the high rates of arrest of G1 and G2 that was seen in the cells treated with e-liquid in the previous study by the Yu et al. So the reduction of the plant height might also due to the decrease in mitotic index and the presence of chromosomal aberrations in the dividing cells. The next one is the effect on the mung bean root growth rate. So in 24 and 48 hours, a significant decreasing trend was recorded in flavored e-liquid at concentration of 10% to 15%. And contrastingly, in flavorless treatment, significant reduction in root growth rate was only seen when the concentration of e-liquid was increased to 25%. So the reduction of the root growth rate is resulted from the reduction of the mitotic index, which indicate the inhibition of the cell division. And this can be considered as indicators of toxicity, which is a general phenomenon caused by the most toxic chemicals. Moving on to the effect on the percentage of mitotic index, we can see that there was no significant difference among means after 0% concentration in both flavors and time exposure treatments. So it can be said that both flavored and flavorless e-liquids can be considered in the lethal range since the mitotic index is reduced by 50%. This implies both flavors have a strong cytotoxic impact on the mung bean. The cytotoxic chemicals in the e-liquids may interfere with the normal process of mitosis, hence inhibiting the synthesis of DNA and also delaying the cell cycle phases. So the last parameters observed was the chromosomal aberration. So similarly with the previous parameters, there was also no significant difference among means after 0% concentration in both flavors and time exposure treatments. So these are all the chromosomal aberrations found in the mung bean root tip cells. The first one is the chromosomal thickness, and this is classed as serious because most of the abnormal chromosomes found were clumped. And chromosomal thickness caused by changes in specific non-histone protein whose function is required for chromatic separation and segregation. The second one is the C mitosis. So the occurrence of the C mitosis is due to the effect of chemical toxin on the spindle fibers and the turbogenic events. And the last one is the chromosomal bridges. So these abnormalities are formed by the chromosome stickiness, which then prevent full separation during anaphase, indicating a potential mutagenesis event in the cell. So in conclusion, flavored and flavorless e-cigarette liquids were both confirmed to be cytotoxic and genotoxic in comparison with control, as they greatly affect all the parameters. Flavored e-liquid severely affects the mean plant growth rate, plant height, and root growth rate compared to the flavorless e-liquid. And finally, studies should be considered to determine the underlying causes of the differences in genotoxic and cytotoxic potential of the e-cigarette liquid samples. This is all the references that I use in the thesis. That is all. Thank you.
Okay, so we open for the question for the panel.
Okay, Akila.
Yeah.
Through your literature search, what are the examples of agents that are present in the e-liquid in the e-cigarette?
The most common mutagen are found mostly are heavy metals. Heavy metals and
only heavy metals.
Heavy metals, diacetyl compound, which can cause severe respiratory disease, specifically in the vanilla flavor, and also aldehyde compound that can also cause a severe respiratory disease too.
Okay, thank you.
Okay, any question? Yeah, so Akila, what is your main finding?
So my main finding is that both the e-liquids, regardless of the flavor, both are cytotoxic and genotoxic to the mung bean, which means that it might be also toxic to human who consumed the e-cigarette.
Okay, so tak bagus lah dua-dua pun kan. Am I right? Okay. Any question? Time? Dah cukup 10 minit Puan Fi? Belum?
Baru je bunyi.
Okay.
10 minit.
Alright, so thank you Akila.
Thank you.
So can we move to the next presenter, Nurul Anis binti Abdan. Anis ada?
Ada.
Ada. So Anis ready?
Sekejap ya saya share.
Okay.
Can you see my slide?
Yes, can.
Okay. I will start now.
Alright. Yes, you may.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good morning I bid to all the panels and my friends. My name is Nurul Anis binti Abdan and today I would like to share my review on different surface sterilant used towards 5 selected bamboo species. In this review, I will cover on the introduction, research question, aim of study, and the contents of this review, which are plant tissue culture, sterilization method, surface sterilant, bamboo species, my analysis of study, and the conclusion and recommendation. Bamboo is a giant woody grass with a variety of purposes, and it is widely used as a raw material in industry. In handicraft, its fiber are used to weave clothes and make paper, and its shoot and sprout can be consumed. Due to that, the demand of bamboo is increasing, lead to deforestation. To encounter this problem, the traditional propagation and micropropagation is the solution that we need to maintain its survival. However, traditional propagation is not suitable for large-scale plantation due to several limitations. Therefore, micropropagation of bamboo is more likely having a huge advantage due to its capability in raising large number of identical plant species in a very short period of time. However, micropropagation of bamboo required a septic environment, thus the usage of disinfectant is needed to make the process succeed. These are the research questions. What are the major problems related to bamboo species? What is the contaminant that usually invade the bamboo species? What is the possible route of contamination to enter the explant? Which type of the explant that usually used by researchers? And which are the surface sterilant recommended by majority of studies? Thus, my aim of study are to highlight the usage of several surface sterilant on 5 selected bamboo species and also to determine the appropriate surface sterilant for sterilization of various bamboo species during micropropagation. Plant tissue culture, or also known as micropropagation, is a technique to propagate the plant by in vitro, which means the process is performed in the test tube or culture dish that contain nutrient media. Due to advanced research, plant tissue culture protocols serve varied purposes like biological activity, improvement of plant growth, secondary metabolite production, and medicine production. Beside here is the process of the plant tissue culture protocol. Here is the most important part in plant tissue culture, where it needs to be done under a septic environment because in vitro culture only depend on the nutrient media for their survival. There are two possible routes of contamination, which are from the mother plant itself and due to inappropriate operator handling. These are the most common type of sterilization methods. For the sterilization of the explant, it is suitable to use chemicals. These are the chemicals that mainly used by researchers in sterilizing their explant. We can see here there's a lot of surface sterilant to be chosen. Therefore, we need to find which one is efficient in reducing the contamination of bamboo. These are the five bamboo species that I choose for this review. All of these bamboo are originate from Southeast Asia, mainly in Malaysia, and it is a sympodial type of bamboo. Some of these bamboo are commercially used to make a variety of products. For the analysis of study, I will show the type of contaminant reported on 5 bamboo species, the explant type, and the surface sterilant used. It was reported that Bambusa vulgaris was invaded by Pseudomonas oryzae, which is the fungus that caused bamboo blight disease in Bangladesh, India. The researchers recommend to use the combination of fungicide like carbendazim and mancozeb or carbendazim and phytolan to eradicate this fungus. There is study on sterilizing the seed and nodal segment of this bamboo using sodium hypochlorite and mercury chloride respectively. On Schizostachys brachydactylum, it was found a new fungi called Archidium bambusicola. It was found in the dead gum of this bamboo. From another study, the sterilization of this bamboo using seed as an explant and was surface sterilized by sodium hypochlorite. Witch's broom disease, which is caused by Epicloe bambusae that invade the Dendrocalamus asper tree in Dendrocalamus plant in Indonesia, and also Phytoplasmas, which is an obligate bacterial parasite, invade the Dendrocalamus asper plant, showing the chlorosis symptom. There are several studies using seed and nodal segment of this bamboo. The treatment of seed is also same like the Schizostachys brachydactylum and Bambusa vulgaris, and the nodal segment requires several surface sterilants such as commercial detergent like Tipul and the antibacterial agent like streptomycin and tetracycline, and fungicide like bevistine and also mercury chloride. On the Gigantochlora scottechini and Gigantochlora levis, it was reported that the fungi that invade these two bamboo is such as Colelotrichum species that cause the leaf spot disease and Aciculosporium species that cause the witch's broom disease. The Mohanan 2017 recommend to use fungicide like Tiram, Benlate, Captain, Carbendazim, and Triazolone to eradicate this fungi. There was no study reported on sterilization of these two bamboo. The nodal segment requires several surface sterilant rather than seed. It is because large intercellular space in nodal segment are exposed during cutting prior to sterilization of explant, allowing the bacterial and fungal spore to enter the sample. Therefore, to counter this problem, several chemicals are needed. And sodium hypochlorite is known to be less hazardous chemical than mercury chloride. Therefore, it is a suitable solvent to be used on seed, which is more delicate than nodal segment. The nodal segment is more robust in structure, therefore mercury chloride can be used to eliminate this microbe. Mercury chloride also proven effective by majority of study and is recommended to use in sterilization process in various bamboo species. And as stated from the table, the fungicide carbendazim are often recommended by researchers to eliminate the fungal contamination. Therefore, in conclusion, the contaminant reportedly leads to various diseases on bamboo species, and nodal segment from mature gum of bamboo species require more surface sterilants because the mature gum has high risk of having endogenous contamination. Sodium hypochlorite is preferred in sterilizing the seed, and mercury chloride is preferred in sterilizing the nodal segment of bamboo. And carbendazim is the preferred fungicide to eliminate the fungi. For the recommendation, more studies should be done and focus on explant disinfection towards bamboo species, and the future study need to be precise in adding more information about bamboo disease and the method to combat the disease. With that, I end my presentation. Thank you for listening.
Okay, thank you, Anis. So we open for Q&A.
Anis, I have a question based on your literature. Does it seems that the pathogen that mostly infected bamboos, does it belongs to bacteria, fungi, or other like parasites?
Usually, the most contaminant that reported infect the bamboo species is fungi because fungi can. Because bamboo reside in the forest where it was exposed by a range of climatic condition. Therefore, the fungi is preferred to invade the bamboo species rather than bacteria.
Okay, and based on your last conclusion, I think, can you go to your conclusion?
Okay.
Okay, you said that endogenous contamination. So can you please explain, especially by fungi?
Okay. The bamboo species may contain the endogenous contamination. It is microbes that stay dormant inside the bamboo, which means it can be stay dormant in the extended period of time, like when we cutting the nodal segment, the fungal spore may enter the bamboo. Therefore, we need several surface sterilant to eradicate the endogenous contamination also.
So meaning that the endogenous is more like more not something that is to invade and cause more risk as compared to the one on the surface only, right? Because endogenous is the one that enters. Because this one is what Ernie studied before about endophytes, right?
Yes.
So is it more more difficult for it to be eliminated?
Yes, because sometimes the endogenous microbes can be very hard to eliminate. So maybe we need to use antibiotic or a stronger fungicide to eliminate these microbes.
Okay, thank you.
Thank you.
Okay, thank you. So this is the sixth presenter ke? Presenter berapa?
Fifth, because Shabika didn't present. It's the fifth.
Okay, so after next is Izzati Nasuha. Alright. So after Izzati Nasuha, can we have a 10 minutes break? Setuju tak?
Setuju.
Yeah. Okay. So Izzati Nasuha, you can present your presentation now. Izzati Nasuha ada tak? Izzati, Izzati, ada. Boleh. Boleh present ke sekarang?
Boleh kejap
Okay, you can share.
Boleh nampak ke?
Okay, boleh. Tapi you tukar ke slide presentation. You tukar ke slide presentation.
Ready.
Tengah loading lagi dah. Okay, kita tunggu.
Assalamualaikum and good morning to lecturers and my friends. My name is Izzati Nasuha binti Muhammad Samsidi under supervision of Dr. Muhammad Fakar Zaman Beraja Yahya. The title that I want to present today is the domain analysis of uncharacterized protein in Salmonella typhimurium. Salmonella typhimurium is the gram-negative bacteria that lives in the intestinal tract of animals and can cause foodborne disease called salmonellosis. Salmonella typhimurium can cause both gastroenteritis and invasive disease, and in some cases can cause fatality. The symptoms of this infection is fever, nausea, abdominal pain, and diarrhea. In the previous study, it was recorded around 93.8 million cases for Salmonella infection and 80.3 million from it was accounted for foodborne. So the objective of the study is to analyze the functional domain of uncharacterized protein in Salmonella typhimurium by using BuzzFeed and Concept Domain Database CDD. The method that I use in this study involves 4 databases, which is UniProt KB, BuzzFeed, CDD, and Cello2Go server. The protein sequence of the uncharacterized protein sequence of Salmonella typhimurium, The molecular function, the biological pathway was retrieved using UniProt KB database. The subcellular localization was retrieved using Cellotogo server, and the domain classification of the protein was retrieved using conserved domain database. The uncharacterized protein of Salmonella typhimurium was analyzed using BLAST-P and CDD, and all the data retrieved was recorded in the Excel and summarized using pie chart and table. The figure 1 shows the classification of biological pathway. As you can see, the biosynthesis pathway hold the highest percentage in the biological pathway, followed by transcription and metabolism. Biosynthesis pathway includes peptidoglycan biosynthesis, protein biosynthesis, amino acid biosynthesis, and lipid biosynthesis. And for the metabolism, includes signal transduction, DNA metabolic process, and catabolic process. Transcription was occurred mostly in DNA activity. Next, we go through to the classification of molecular function. DNA binding has the highest percentage in the molecular function, followed by ATP binding and metal binding. Binding protein was recorded as the most abundant for the function in Salmonella typhimurium, which also includes DNA binding and metal binding in the previous study of SAR et 2020. Next, the classification of subcellular localization. The highest is cytoplasmic, and it is in compliance with the previous study by Yaya et al. 2017, which also recorded the cytoplasmic as the highest percentage for subcellular localization in Salmonella typhimurium. Figure 4 shows the classification of protein domain in Salmonella typhimurium. Fish-related domain was the highest, followed by autotransporter domain after the unknown function. Fish-related domains such as fish C2 consists of fish regulatory protein C2 that has a function as DNA binding and involved in the lysogeny process. For the autotransporter domains such as auto-BA domain has a C-terminal outer membrane beta barrel which protein diffused through it. Protein being transported through it. And as you can see in this table, the molecular function of the autotransporter domain is a porine activity. Porine is a beta barrel protein which help the molecules diffuse through it. And for the fish C2 is also the NA binding. In conclusion, the biological pathway, subcellular localization, is important in subcellular localization. Biological pathway and molecular function is important in determining the function of the protein, and the domain analysis of Salmonella typhimurium. It was attained using UniProt KB, BLAST-P, CDD, and Cellotogo. The highest protein domain in this study is the fish-related domain, and it's play a key role in the lysogeny process. The project is recommended to be further studied to have a better understanding about the relationship between protein domain and its function. With that, I end my presentation. Thank you.
Okay, thank you, Izati. So I open for Q&A. Any question from the panel? Okay,
Izati,
okay,
Yes, madam.
what is the significance of looking at uncharacterized proteins in Salmonella specifically?
Sorry, madam, but I can hear it clearly. Can you
What
repeat?
is the significance of looking at these uncharacterized proteins in Salmonella? What are you looking for?
What I'm looking for in this study is the protein, the domain of the protein.
Why is the domain important?
The domain provide insight of the structure, the sequence, and also the function of the protein itself, so that we can know the function of the protein and helps in the drug in the that can help in the that can help the researchers to produce drug to help in this salmonella disease.
Okay.
Alright. So any question? All the time is already.
Okay, time's up for 10
Time's
minutes.
up already. Okay. Alright. So thanks, Izati. So for that, we break for 10 minutes. Okay. We will meet again about 10:28. Okay. So kita break sekejap lah. Thank you.
Okay, saya record.
Alright. So Nurlana. Okay. So you can start your presentation once we are ready.
Alright. Bismillahirrahmanirrahim. Assalamualaikum and very good morning. I bid to the panels and my fellow friends. My name is Nurliana Bati Salihuddin and I'm going to present my final year project. Without further ado, we're moving into the introduction. As we all familiar with DNA extraction, is a process for isolating genomic DNA that carries genetic information out from the nucleus of organism cells. In this study, we focus on the mature leaves because there are some obstacles obtained from the young leaf, which are then not available for throughout the year, especially among the series 3. And young leaf also unclear about the mutation characteristic. And next, they are prone sensitive to temperature fluctuation that cause accumulation of secondary metabolites. And last but not least, it is also incompatible for long storage when collected for distant places. Therefore, scientists use a mature leaf instead. Getting a pure and high yield of acetate DNA becoming a limiting factor among the plant cells because of two contributing factors. The first one is the formation of tough cell wall of plant cells, and the second one is the production of secondary metabolites by the plants. In this significant of review is to provide some ideas for researchers to optimize their DNA extraction protocol against difficulties from mature leaves. Here is some research question successfully extract for this review, and this is the objective being listed out for answering the research question. So we're moving to the first point is the principle of DNA extraction protocol.
Yang slide tu tak tunjuk the objective. It's still on the problem statement.
Not moving.
Oh really?
Yeah. We start at the problem statement slide.
Is it still stuck?
I'm not sure. Have you moved your slide?
Yeah, I moved my slide.
But it's not moving in the. Okay, now research question.
Right now it's research question.
Yes. Is significant of review?
Oh, it's quite stuck.
Okay.
Because I'm known as objective.
Objective, yeah? Okay, so it's objective already.
Okay. Right now is the principle of DNA extraction protocol.
Not yet. It's still on objective.
Is it still on objective?
Yes, it's
still on objective.
Oh.
Making one.
If you cannot move, sometimes we convert to PDF and then it can move. I don't know, another alternative.
Or else just no need to guna use this one slideshow. Just kalau this one.
This one, not the slideshow.
Okay, can you try to go to the next slide?
I'm going to the next slide. Is it?
Oh, not moving also. It's still on objective.
Not moving as well.
Doktor.
Tak apa.
Tak apa, pelan-pelan. So now objective.
So sorry, Doktor.
Okay, tak apa. So now you dah move ke the next slide
Yeah, it's on principle of DNA extraction protocol.
Kalau tak buat slideshow pun sangkut tak ke?
Betul lah.
Yes.
Tak buat slideshow. Sis sangkut ni. Dia macam stuck on the objective slide.
Maybe we can proceed with the next student. We kena tukarkan dulu pada PDF and then present balik after this.
Oh, alright.
Boleh
Alright, sure,
okay.
Doktor.
Alright. Okay, thanks. Okay, so kita proceed with the next speaker lah, Liana. After this, I will call you back. Once you're ready,
Alright, Doktor. I'm so sorry.
Alright, no mind. No Atika? No Atika, are you ready? No Atira, sorry. No Atira Izzati.
Yes, Doctor.
Are you ready?
Yes.
Okay, good. Alright, so you can share your presentation slide and then you can start present once you're ready.
Okay. Can you see my slide, Doctor?
Yes, but not in the presentation mode yet. Okay, this is in the presentation. Okay, yes.
I will start now.
Oh, yes. Repeat, ready?
Yes.
Alright, so you may start.
Assalamualaikum warahmatullahi wabarakatuh to all examiner and my fellow presenter. My name is Nur Atira Izzati binti Muhammad Ali under Dr. Syamsul Bahrin bin Gulam Ali. Today, I will present about the impact of malaria and filariasis concomitant infection. Co-infection is defined as the simultaneous infection by different parasites independently on a single host. Currently, many research are looking at the impact of co-infection as they may have serious consequences, especially for human and animal health. In the past few decades, there has been increasing interest in the study of helminth and malaria co-infection. Several researchers have found an association between malaria and lymphatic filariasis infection. It is believed that the same vector can transmit both diseases and they can co-occur within the human host, especially in areas where both parasites coexist. This slide shows 5 research questions about this review, which are: what is the relationship between malaria and lymphatic filariasis infection? Second, what is the impact of malaria and lymphatic filariasis co-infection on the host immunity? Third, what will happen to the individual if multiple parasites reside in them? Fourth, how malaria and lymphatic filariasis infection can be transmitted to human hosts? And lastly, what is the suggestion and recommendation from this review? Therefore, this review aims to provide an overview of lymphatic filariasis and malaria infection induced by helminth and protozoa parasite, emphasizing the vector responsible for transmitting both diseases and the effect of concurrent infection on the host. Lymphatic filariasis is a parasitic disease caused by helminth. The filarial nematode that induce the illness are Wuchereria bancrofti, Bugia malayi, and Bugia tibori. However, among those 3 parasites, infection with Wuchereria bancrofti is the most prevalent in humans. Even though Wuchereria bancrofti infection is not fatal, they contribute to 90% of all human lymphatic filariasis infection. Lymphatic filariasis also associated with severe pathologic disease outcome known as elephantiasis. This is the example of elephantiasis, where the condition of enlargement of the leg due to the accumulation of the fluid. Malaria is a mosquito-borne disease that is caused by protozoan parasite from the genus Plasmodium. Humans can be infected by 4 malaria parasites, which are Plasmodium falciparum, Plasmodium malariae,
Plasmodium vivax, and Plasmodium ovale. In addition, Plasmodium knowlesi can also infect humans, which in previous study usually use MECA as its natural reservoir. Malaria and lymphatic filariasis is a mosquito-borne parasitic disease. Both infections are transmissible by the same vector species, which is Anopheles mosquito, and both have no or almost no host other than human. This is the life cycle of malaria and lymphatic filariasis. The thing that differentiates between both infections are their definitive host and intermediate host. The definitive host for malaria is female Anopheles mosquito, while for filaria is human. The intermediate host for malaria is human, while for filaria is female Anopheles mosquito. Malaria and filaria cannot be transmitted directly from person to person. However, this infection is favored by vector. In this case, is mosquito. Mosquito from the genus Anopheles are the ones that usually transmit parasite that cause human lymphatic filariasis and malaria infection. The impact of co-infection. Multiple infection on a single host will significantly alter the clinical manifestation, making the diagnosis process more difficult. Since both infections may have similar clinical presentation, this will result in an incorrect diagnosis. The treatment for co-infection also can differ from those mono-infection. Therefore, a poor diagnosis may delay implementing the appropriate treatment, which lead to fatal. Immunology related to both pathogens. Generally, immunity to helminth and protozoa infection are being associated with helper T cell, also known as Th cell. Protozoa is correlated with Th1 immune response, meanwhile helminth being associated with Th2 immune response. To protect the host from both infections, these immune response are required. The impact of malaria and filaria co-infection on the host immunity. Malaria immunity is influenced by helminth infection. It is believed that when co-infection occurs, the Th1 response is suppressed in the presence of the Th2 effector response. This means that helminth can suppress immunity to Plasmodium species. Helminth infection have been reported to aggravate the symptom of malaria through blood loss, which lead to anemia and impairing the host's ability to mount the Th1 type immune response. So, as a result, the human host might be unable to counteract the inflammatory response to malaria. It will take more time to eliminate, kill, destroy the malaria pathogen due to the less Th1 response. In early study, malaria and lymphatic filariasis control strategy are independent of each other and target each infection individually. However, World Health Organization is implementing new strategy that target both diseases simultaneously, known as integrated vector management. The majority of vector control is currently carried out by the Rollback Malaria Partnership (RBM) and the Global Program for Elimination of Lymphatic Filariasis (GPELF). They target Anopheles vector of malaria, in which its action also reduce the transmission of filaria. This review clearly indicates that there is an association between malaria and filaria infection, since both can be transmitted by the same Anopheles vector. Although the simultaneous transmission of both parasites by the individual vector is rare in nature, concurrent malaria and lymphatic filariasis infection can still occur if both parasite prevalence is high. Persistent helminth infection in the host can significantly alter the susceptibility of the host to protection from Plasmodium. Therefore, understanding interaction among both parasites, especially where their co-endemic is critical to successfully execute elimination and control program for malaria and lymphatic filariasis. I will end my presentation with some suggestions and recommendations from this review, which are implementing public engagement program for mosquito-borne infection, prevention and control, and creating a new diagnostic test that can detect both infections in a single test format. Here are some references that I used in my presentation. So that's all from me. Thank you.
Okay, thank you, Atira. Atira. So I open for Q&A from the panel.
Atira,
Yeah.
through your literature review, in Malaysia, how serious is the co-infection of malaria and filariasis?
Through my literature review, Malaysia does not have many cases about co-infection with both parasites. It's only a small number.
Why is that so?
Because in Malaysia, because co-infection is commonly occur between Wuchereria bancrofti and Plasmodium falciparum. We are famous with Plasmodium knowlesi. So co-infection not always occur in Malaysia.
Okay, thank you.
Okay. How about time, Bafi? Dah due or not?
1 more minute.
So any question? Your objective, can you show again your objective, Atira? Do you have your objective here?
Yes.
To provide knowledge of lymphatic policies, infection. So did you achieve your objective for this LR?
Yes.
For the review?
Yes.
Yes, right? Okay, so any question? Any more question? No? I think if there's no questions, so we will end here. Okay, thank you, Natira.
Thank you.
Okay, so we move to the next presenter. Liana, are you okay? Liana dah okay ke? Nurul Liana?
Yes, Doctor. Saya dah save ke PDF.
Alright.
But I think my internet is quite low. It's quite low, but I think I can do it.
Okay, so you want to try?
Yes.
Okay, so you can share your presentation. Alright, okay, Liana.
Doctor can see my slide?
Yes, can.
So I was, is it moving?
Yes, it's moving now.
Alright.
Okay.
So I start. Bismillahirrahmanirrahim. Assalamualaikum. I bid to panels and my fellow friends. My name is Nuruliana and I'm going to present my final year project. Without further ado, we move to the introductions. As we are familiar with DNA accession terms, is a process of isolating the genomic DNA carries genetic information out of the nucleus of an organism cells. In this study, we focus on the mature leaf, where there are some obstacles came from the young leaf, as they are not always available throughout the year, especially among deciduous trees. And second, their mutation characteristic is still unclear to study genetic variations. Young leaf also more sensitive to temperature fluctuation, causing an abnormal accumulation of secondary metabolites. Lastly, they are compatible for long storage when we collect samples from distant places, and therefore scientists use a mature leaf instead. Getting a pure with high yield or extracted DNA becomes a limiting factor among plants due to two contributing factors. The first one is formation of tough cell walls in plant cells, and the second one is the production of secondary metabolites by the plants. This significance of review is to provide some ideas for the researchers to optimize their DNA accession protocol against the difficulties from mature leaves. Here are some research questions that extract for the review, and this is the objective answer to the research questions. So let's start with the first point, is the principle of DNA accession protocol, and there are three central steps. The first one is the cell lysis, involve two techniques, which is the physical and also the chemical. The physicals, we use the liquid nitrogen during the grinding process to break down the tissue into the powdered material. Meanwhile, the chemicals involve the accession buffer, which aid in dissolving the lipids and proteins of the phospholipid bilayers and thus release the DNA. Second one is the separation of DNA from impurities, which also require two techniques, is the partitioning and also precipitating. Partitioning involve the organic solvents, as is aid in separating a liquid phase, as we can see in the picture, DNA in the aqueous, and meanwhile the cellular debris in organic solvents. Precipitating was done by salt and alcohol, as is precipitated DNA from the aqueous. Last but not least is the preservation, where a liquid nitrogen and chelating agent will deactivate endonuclease and minimize DNA shearing. Therefore, we can maintain the DNA integrity. Moving to the next point is the mature leaf remarkable attributes over the young leaves. The first attribute is the rigid cell walls, where the mature leaf contain a new layers called rigid secondary cell walls, which will double the matrix and thickness of the cell, of the tissue. Meanwhile, the young leaves only contain the primary cell walls. The second attribute is secondary metabolites, where the mature leaf require high in concentration used for chemical defense for survival. Meanwhile, the young only possess less secondary metabolite because they use only aggregate for early growth instead of defense. Moving to the next point is the modification in selected protocol. Based on the previous studies, utilization of mature tissue, there are several modifications made, which are the addition of chemical, concentration of reagents, repetition in step, duration of incubation, and also the temperature. So we start with the addition of chemical. It involves two, which is a pre-extraction buffer and also the liquid nitrogen. In pre-extraction buffer, there are three types of buffer, which mostly eliminate the most secondary compounds. Meanwhile, for liquid nitrogen used in most studies, help facilitate breakdown the mature plant cells containing thick and also rigid cell walls. Moving to the concentration of reagent, involve a CTAB buffer. Increase in CTAB and also sodium chloride increase the efficiency of the lysis and also the precipitation process. So does the PVP and also turmeric ethanol, which mostly deal with the polyphenols contamination. But recent study from Kuma in 2018 said that 2.8% of PVP giving the best result, consider that too high in PVP concentration could lead to low yield of extracted DNA. Moving is the duration of incubation during extraction buffer and also alcohol precipitation. Increase in duration will increase the DNA extraction process in both technique. Moving is the repetition in step, involve a washing, which the chloroform isoamyl alcohol washing, it will help to remove polysaccharide and also strong protein contamination. Meanwhile, the alcohol washing will help eliminate remaining salt contaminants. Last but not least is the temperature modification, where study in Kuma said that 65 degrees Celsius during a extraction buffer giving the best result. Meanwhile, Suba in 2014 said that 4 degrees Celsius of alcohol precipitation giving the best result compared to negative 20 or the room temperature. So moving to the last point is the quality and quantity analysis of extracted DNA. Both quality and quantity control use the spectrophotometry and also gel electrophoresis. In spectrophotometry, in quality control, require 2 absorbance ratio given a standard range. If any reading outside the range, it considered DNA extracted containing a contamination. Meanwhile, for the gel electrophoresis, we can see a clear, intact, and absence of smear of band visualization indicate a good quality. As we can see in the table 1, is the previous study that utilized a mature leaf and used a selected modified protocol and successfully obtained around 1.7 to 2.0, considered is acceptable for a good range of quality of DNA. And this is a figure 1, shows a comparison of DNA bands on different approach. We can see with modification, it is more clear, less smear, and no RNA contamination. Meanwhile, for quantity control, the DNA concentration can be obtained from the formula, where the spectrophotometry using the reading of 260. Meanwhile, for gel electrophoresis, comparing the band intensity with DNA marker. Study by Fatima et al. Shows the comparison of quantity of DNA. We can see that the significant difference when using a modification, you obtain a quite high quantity of DNA compared without the modification. So in conclusion, DNA extraction, the extraction of DNA from mature leaf can obtain with several modifications, but we need to understand the internal component of the plant of interest itself, and we need to optimize our modification until we get the quality and quantity of selected DNA. And not to forget, we need to analyze the quality and quantity assessment of selected DNA in both spectrophotometry and also the gel electrophoresis. And with that, I end my presentations. Thank you.
Okay, thank you, Diana. So I open for the question.
Okay, Assalamualaikum.
Waalaikumsalam.
Liana, sorry. Can you please go back to your just scroll. A little bit more. Okay, this one.
This one?
Concentration of reagent.
Right.
Okay, you have been using CTEP buffer. So, an addition of sodium chloride.
Yes.
So, is this the modification that you mentioned in the last graph that shows that high quality DNA has been obtained after modification of the CTEP buffer?
Yes. The study, the previous graph said the study from Fatima et al. They use for the CTEP concentration of CTEP buffer, they use a high concentration for both for CTEP and also sodium chloride. So therefore, it helps to obtain a good high quantity of DNA extracted.
So what is the concentration of sodium chloride? Because you said increased efficiency of the lysis precipitation. But isn't there a maximum amount that you can add? That if you add that maximum amount, it can cause not in a good way, but you have something in reverse. Meaning that if you add too much, maybe there will be effect on other components of the DNA, for instance.
For CTAB and also the sodium chloride and 2-mercaptol ethanol, there's not quite study about the reverse effect. But for PVP, they did say that if too high in PVP, they're low in yield. But based on our review of this previous study that I've seen, the most significant increase, the most high concentration of CTAB they use is 4%. And for the sodium chloride is also around 3%. But mostly the previous study used around 1.4 to 1.7% concentration is enough for them to obtain a good results.
Okay, last question. For this one is not the one that you skid, right? This is a manual method, right?
Yes.
So how long will it take? Because there is overnight method involved, I think I saw just now.
Yes.
So in terms of the disadvantage, it takes longer, right?
Yes, but it can modify better compared to kit.
Okay, that will be a good reason. Thank you very much.
Alright, thank you so much, Doctor and Madam.
I'm suppose time is already due.
Yes, due.
Okay, ready. Okay, so thank you, Yana. Alright.
Thank you so much, Doctor.
Alright, so we move to the next presenter, Ms. Sarah. Are you ready? Ms. Sarah?
Yes, Doctor.
Okay, so you can share your presentation slide.
While the next presenter get ready, Syafiqah tadi kata she cannot share the video tu dekat dekat ni kan WhatsApp because it's too big. Itulah maybe Syafiqah kan dia boleh letak dekat ni kot, dekat YouTube. Dia ada YouTube channel tak?
Sekejap, dekat Telegram I dah hantar dekat you, dapat tak?
Telegram.
I send to both Fala and you and it's from Dr. Wan.
Yeah, betul.
Okay. Got it.
So maksudnya bila time Syafiqah kita tengok dekat Telegram and then kita suruh dia standby untuk Q&A lah kan?
Boleh.
Kan sebab dia kata dia memang tak boleh.
One of us can present kot dia punya video.
I tak reti daripada Telegram ni.
Sebab komputer I tengah pakai so I can't do that. You just mana
I can try tapi I tak download lagi.
Tak apalah.
Kalau boleh. Tapi kita lepaskan yang lain dulu.
Yes. Okay.
Okay,
Alright.
yeah.
Okay, kita teruskan yang ni dulu Maisara. Okay, Maisara dah ready?
Saya dah nampak ke slide?
Dah nampak dah.
Dah.
Ya.
Thank you.
Okay, so you can start Maisara once you're ready. Okay.
Assalamualaikum warahmatullahi wabarakatuh. I bid to the lecturers and my fellow friends. My name is Maisara binti Muhammad Fisul and supervised by Cik Azrina binti Abdul Karim. Today I want to presenting the research review article entitled Effectiveness of Natural Antibacterial Agents on Klebsiella pneumoniae. Let's take a look into the introduction of this review. Klebsiella pneumoniae is a bacteria that has been considered as a health threat to community worldwide. Antimicrobial agents that is used for antibiotic drugs into the human causes so many side effects. Therefore, we need to discover the alternative findings to inhibit K. pneumoniae growth and their virulence towards the antibiotic by using natural compounds. The objectives and aim of this research is to identify the possible natural antibacterial agents that can act on the K. pneumoniae. Next, to understand the mechanism of the antibacterial agents towards K. pneumoniae, and lastly, to identify the natural antibacterial agents that is compatible with the human body. There are some research questions or gaps that has in this research review. First one, it will be time consuming as the researchers undergo different treatments and the testing process to find the specific active compound in the natural extracts. Next, the treatment option could be limited as the Klebsiella pneumoniae is already resistant into almost available commercial antibiotics. Next, the result obtained from the lab test bacteria in the review cannot be represented into the real life effect. And lastly, the most researchers identify the general compounds of the plant extract as an antibacterial process. However, the question is what factors that make these components can be expressed as antibacterial. The content that I have constructed in this review is the background of Klebsiella pneumoniae, next antibacterial action of plant extract, which is clove plant and essential oil in plant extract, and lastly, the strategies to affect the K. pneumoniae. Now let's take a look into the background of the selected bacteria, Klebsiella pneumoniae. Klebsiella pneumoniae is a gram-negative bacteria that is very resistant towards almost available antibiotics. Therefore, it is emerged as a common multi-drug resistant bacteria, MDR. According to World Health Organization, it is considered, it is in the list of the critical priority list, and the bacteria is urgently need to treat with the new antibiotics. K. pneumoniae also able to colonize in the human gastrointestinal tract, mucosal surfaces, bladder, and skin. Some of the antibacterial action of the plant extracts is due to their active compound. For example, clove plant, Syzygium aromaticum. Syzygium aromaticum has a bioactive component called eugenol. Eugenol is also effective to display antimicrobial effect. It is a high soluble molecule that can engage in the bacterial cell membrane and disturb the hydroxyl group. Then the eugenol can pass through the hydrophilic part in the membrane and penetrate into the important cell components of the bacteria. Next, the group phytoalexin. Group phytoalexin is a newly founded group in the essential oil that can be extracted from the plant. It is proved that it has high antibacterial activity, which is bacteriostatic and bactericidal. Next, it is specifically functions to affect the biofilm formation. Next to this phytoalexin actually produce inadequate amount by plants when they get injuries. For example, the invasion of pathogenic fungi and bacteria. Let's take a look into the oxidative stress action, which is the way how to inhibit the growth of bacteria. The ROS, reactive oxygen species, when it is in the excess or imbalance of ROS can lead into the condition called oxidative stress. ROS is a natural byproduct produced by the cellular respiration of the cell. However, increased ROS can lead into the harmful effects towards the cell. Bacterial membrane can be disrupted by the condition of oxidative stress. Therefore, it can lose a lot of membrane protein. As a result, the bacterial cellular process could be slowed down. Next, the lavender essential oil also can produce ROS. The application of LVO towards the Klebsiella pneumoniae can cause oxidative stress towards the Klebsiella, then cause lipid peroxidation that leads into the membrane disruption. It was found that in the research that K pneumoniae that when treated with LVO, there is an increase of ROS level compared to the non-treated Klebsiella pneumoniae with LVO. It is proof that the exposure of LVO can result in the cell damage of the bacteria. Next, the strategies to inhibit the virulence of bacteria is by the degeneration of the biofilm. Biofilm actually make as a mechanism of the bacteria to survive in harsh environments. It is one of the major resistant mechanism of the bacteria that make it impermeable into any antibiotics. In addition, biofilm is a major problem in clinical settings as they cause most infections, especially they can adhere into the hospital's instruments. For example, of natural antibacterial agents that can inhibit the biofilm formation is Endographis paniculata, A. paniculata. So, based on these two bar chart, it is found out that a minimum concentration of A. paniculata, as much as 250 microgram per ml, is needed to effectively inhibit the growth of K. pneumoniae and biofilm formation. Next is by the suppression of quorum sensing, QS. QS is a mechanism for bacterial communication, including the biofilm formation. In addition, the antibiofilm strategy involved with the QS inhibition. Klebsiella pneumoniae produce auto-inducer acylhomoserine lactones, C6AHL, and it was found that substance named 3-methyl-2,5-H-furanol was 67.38% effective to inhibit the biofilm, and also need as much 15 microgram per ml also could neutralize the effect of the auto-inducer, which is C6AHL. Next, the synergy effect can be used to inhibit the virulence of bacteria by using the combination of nanoparticles combined with essential oils. This combination is capable to decrease evaporation and minimize the degradation of the essential oil because the usage of the essential oil alone is very limited due to the water solubility and less stable.
Time's up. Sambung sikit lagi.
Okay. For example, rosemary and ginger essential oil, when used the combination with rosemary and ginger essential oil, it has the minimal inhibition concentration as much as 156 microgram per ml, and half of the MIC could inhibit the 100% of adenosine MRKD gene that is important for the bacterial growth. The positive charge of chitosan can create a greater contact with negative charge of the bacterial membrane. Thus, it causes membrane degradation and let the intracellular materials from the bacteria will be released from the inside into the outside of the cell. It's proof that this combination could create a synergetic effect when being combined in nanoparticles together. So here is some suggestion and recommendation. It is must be effective in a long term to educate to inhibit the virulence of K. pneumoniae before the bacteria will change due to mutation. Other than that, treatment process must be standardized to achieve the same effectiveness, and further research is needed regarding this issue. Hopefully, this finding might be a precursor for the researchers in order to develop new inventions of natural antibacterial agents. This is the list of references I have used. Thank you so much for listening.
Okay, actually dah almost 10 minutes Dr. Wan.
One question.
It's already 10 minutes dah. Ask the coach lah.
Pada Dr. Wan.
So bagi satu soalan lah. So based on your finding, your review, so between the compound, the isolated compound and the plant extract, okay, in your opinion, which one is better in combating the bacteria growth, and why?
In my opinion, the most effective way to inhibit the growth is by the synergy effect, because it is stated that it is natural, and because the chitosan is a natural compound, chitosan have the positive charge molecule, and it can create a greater impact towards the negatively charged cell membrane. There's the antibacterial agents, which is the essential oil, can be penetrated into the bacterial cell membrane and cause the cell membrane degradation.
Synergy effect Synergy effect, so means that combined with the combination compound.
Yes, with the nanoparticles.
Okay, thank you, Ms. Sarah.
Thank you so much.
Alright, okay, thank you. Okay, so the next one, apa dia? Nur Atikah binti Syahrul Zamri.
Ya, doktor.
Mana dia tu? Suara pilihan dia. Nur Atikah. Okay. Ready?
Dah, saya.
Alright. So you can start. So share your presentation slide, and then you can start once you are ready. Okay.
Can you see
Boleh
my
nampak?
screen, doktor?
Ya, boleh.
Okay. Assalamualaikum and a very good morning to panel of examiners and my fellow friends. My name is Nur Atikah binti Syahrul Zamri under the supervision of Dr. Nora Shirin Muhammad Jamil, and I will present about my review article titled Mode of Action and Imaging Resistance of Macrolides, a review. Firstly, macrolides was first discovered in the 1950s and is characterized by their macrocyclic actin ring. The most famously known macrolides is the erythromycin, which is a 14-membered macrolides that is isolated from soil bacterium Streptomyces erythraeus. Macrolides was mostly effective on gram-positive bacteria and few gram-negative bacteria. They are also used for respiratory, gastrointestinal treatment and to treat skin, soft tissue, and genital tract infections. Next is the structures of known macrolides. The first generation of macrolides are typed in black, and all of them are natural products, while the second generations are ones typed in red, and the red color in the structure shown in the figure are the modification made from erythromycin and are the semi-synthetic derivatives. Next is the research questions for this review. First, is the macrolides mode of action well established? Second, how does ribosig help in understanding the mode of action of macrolides? Third, is macrolides inhibitory effect selective? Fourth, what are the identified resistant mechanisms against macrolide? And lastly, what can be done to understand more about macrolides mode of actions? The objectives of this review are to revisit and study the true mode of action of macrolides and to identify specific and updated bacterial mechanism of resistance against macrolides. Moving on to the content, I will talk about the mode of action of macrolides. Based on this diagram, it is the traditional outlook on macrolides mode of action and is famously known in biology as the plug in the tunnel model or the tunnel plug model. The primary target of macrolides is the 50S subunit of bacterial ribosome. So how does this model work? Firstly, macrolides will bind at a site called the 23S rRNA within the nascent peptide exit tunnel, NPET, which will block the nascent peptide chain carried by peptidyl tRNAs from passing through and leaving the ribosome. Elongation of protein will then be prevented which led to accumulation of peptidyl tRNAs carrying short peptides that ended up with dissociation of them from ribosome, also known as peptidyl tRNAs drop-off. However, new findings show that through ribosig, mode of action of macrolides could be more complicated than we first thought. New research found that macrolides inhibit translation of subset of proteins selectively. Translation arrest occurred as specific sequence referred as macrolide arrest motifs, MEMS, and stalling of ribosome occurred at those specific sites. We will look in details of these points later in the discussion section of this presentation. Next is the mechanism of resistance against macrolides. Firstly is the target modification that is associated with A2058 and A2059 nucleotide and also ribosomal protein L4 and L22. How does this happen? This phenomenon could occur in two ways, which are methylation or mutation. Methylation and mutation could occur on the nucleotide A2058, while mutation occur on nucleotide A2059 and ribosomal protein L4 and L22. This contributed to the macrolide resistance as they will cause a conformational change on the target site, making macrolides unable to bind. Second is the decline in intracellular concentration associated with efflux pump subfamilies. MEF from Major Facilitator Superfamily MFS, which are MEF-A and MEF-E, and MSR from ATP binding cassette family ABC, which are MSRA, MSRB, and MSRD. These are the known efflux pump that confers resistance of macrolides by extrusion of the macrolides molecule from bacterial cell. Third is the macrolide modification made by specific enzymes known as macrolide phosphotransferases, MPH, and macrolide esterase ARES that cause macrolide unable to bind to their target site. The last but a fairly new mechanism is the target protection by ATP binding cassette protein like MSRE, in which it can insert a needle-like domain to location where macrolides bind and push the antibiotic out of its binding site. Now, in regards to new findings stated earlier, a study by Kanan shows that E. coli that is treated with a 100-fold minimal inhibitory concentration still have approximately 6% protein synthesized, while telithromycin around 25% protein synthesized, and a study by Al-Mutairi shows a 40% protein synthesized for picromycin. This is a proof that while macrolides does inhibit protein biosynthesis, there are still some proteins that it allows to be synthesized. Furthermore, we can see from this diagram that there is a stark difference between control erythromycin and telithromycin for each of these genes. Translation arrest occurred at specific sites are dictated by the high peaks of the graphs. This could relate to the macrolide arrest motifs, MEMS, mentioned earlier, as ribosome will stall when they encounter these unique motifs, which is what led to the translation arrest. As ribosome stalls, the ribosome density at those specific sites will be high, which could be detected through the ribosig technology. It can be seen here that for the METG gene, early translation arrest occurred, whereas translation arrest at inner codon occurred for his P gene. However, translation occurs throughout the GLO-A gene, and the sites in these genes differs based on the drug they were treated with. All these data prove that all proteins are inhibited by macrolides. To conclude, macrolides inhibit production of proteins selectively instead of being a global inhibitor for all proteins. So we can consider macrolides to be context-specific. Henceforth, developing detailed study or experiments to identify true mode of action of macrolides is imperative to first enable study of modifying the macrolides antibiotics to increase its effectiveness, and second, to curb the emerging resistance towards macrolides. From this review, there are still more to know about macrolides despite the many studies done before. As suggestions that may help in further research is by using the combination of mutation study, protein structure study, bioinformatics study, and the whole genome sequencing that to learn more of macrolides mode of action. These are my list of references. So that will be all from me. Thank you.
Okay, thank you, Natika. So I open for question from the panels. Okay, Atika, I just wonder what is the importance of this macrolide actually?
The importance of
Ah.
this macrolides is an antibiotic that is mostly used to treat infections like the genital tract infections and the respiratory treatment I mentioned earlier in the introduction. So and it is mostly used for outpatient settings in a hospital. So without macrolides, there will be more patients that need to be warded in the hospital and could burden the healthcare system.
So normally where can we obtain this macrolide? Bacteria?
The first from erythromycin is the first macrolides isolated, which is from bacteria. But the second generation and the third generations are the modification made from the first generation and other semi-synthetic derivatives. So instead of bacteria, we can make modification from the earlier generation of macrolides instead.
So is there possible to obtain it from the plant? No.
I'm sorry, doctor?
Do you come across that can we obtain this erythromycin from the plant extract?
No, from my readings, I haven't found erythromycin that is isolated from plants.
Oh, okay, I see. Any question from the panel or is the time is due already for me? Then the panel.
Satu je.
Okay.
Why is it that gram-negative ni are not as sensitive to microlides compared to gram-positive?
Because gram-negative has the outer membrane layer as compared to gram-positive bacteria, and because most efflux pump is more on gram-negative rather than gram-positive.
Okay, siapa microbiology lecturer awak?
I'm sorry?
Siapa microbiology lecturer awak? I teach you?
Yes.
Okay. Dah sebut sekarang.
Okay.
Okay, so I think this. Kepala ada soalan kepala?
Tak ada.
Okay, so I think that's all for Atika. Thank you, Atika. So we move to the next one. Ifa. Ifa.
Yes, doctor.
You ready? Atika, can you stop design? Okay, thank you. Alright, Ifa, you can start share your presentation.
Wait a minute. Can you see my slide, doctor?
Yes, can. But please change to slide presentation, slideshow.
Okay, jap. Can you see it's moving?
Right? Yes.
Can you hear my voice clearly?
Yes, can. Alright.
Crystal clear.
So Ifa, you can start once you are ready. So Poppy, okay? Can we?
Okay.
Alright.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good morning. I greet to all of the lecturers and my friends. So I'm Nurul Ifa binti Hashim and today I'm going to present my final year project entitled Bioremediation of Hydrocarbon Contaminated Soil with Pseudomonas species. Hydrocarbon is a chemical organic compound which form the basis of crude oil. Crude oil, or also known as petroleum, is highly effective as a fuel source and has been employed for lubrication and power generation. Due to the high demand of petroleum until today, it caused soil contamination has occurred as a result from the harmful hydrocarbon content in the crude oil. A study by Abdullah et al. Said that it could cause lower rate of growth as well as reduction in plant germination rate. Other than that, it also caused adverse health effects towards humans such as respiratory tract irritation, skin and kidney problem, as well as blood profile disturbance. Next is the treatment available for contaminated soil includes physical separation as well as chemical degradation. However, this existing method tends to leave residues which have high toxicity level, which is why a process that involves the usage of microorganisms to degrade the pollutant are more preferable, and this biological method is known as bioremediation. Some of the potential microorganisms that has been applied in bioremediation includes from the genera Acinetobacter, Enterobacter, and many more. Next is the list of research questions. For my first research questions, as hydrocarbon contaminated soil possess great danger towards organism, it must be treated so that the soil can be reused safely in the farming application of agricultural industry, as well as to reduce health threat towards human who may accidentally consume the contaminated plant. For my next research questions, as being stated by Ojiwumi et al., the existing method tend to leave residues which has high toxicity level compared to the oil itself, which is why bioremediation is preferred as it is environmental friendly, has comparative effectiveness, and does not require much cost. For my next research questions, from my article review, there are 5 Pseudomonas species that were discovered to aid in the bioremediation of hydrocarbon contaminated soil. And some of the species also possess various type of strains. And all of these Pseudomonas species will be shown in table 1 later. For my next research questions, generally, the mechanism used by hydrocarbon degrading microorganism includes emulsification by biosurfactants, adsorption, enzymatic reaction, and eventually the degradation of hydrocarbon. This mechanism is also applied to Pseudomonas species as well, but with a specific biosurfactant known as rhamnolipid. So for my last research question, bioremediation consists of 3 techniques, and these 3 techniques could come out with different results depends on the presence of nutrient as well as adjustment of parameters such as pH and temperature. Other than that, production of biosurfactants could also be considered as a specific factor since it plays a important role during the mechanism of hydrocarbon degradation. So as there are many microorganisms that has been applied in bioremediation, this review article aimed to highlight the availability of several Pseudomonas species that are potential in the bioremediation of hydrocarbon contaminated soil. Moving on to the content of main body, so bioremediation consists of 3 techniques, which is the first one is known as bioaugmentation. It is the addition of exogenous microorganism to the contaminated site, and usually the addition of microorganism depends on the pollutants that needs to be degraded. Next one is the biostimulation, which is the amendment of nutrients such as nitrogen to the residential microbial flora that are already present on the bioremediation site. And lastly is the combination of both, where besides microorganism is being added, nutrient is also being supplied. Next is the Pseudomonas species that has been discovered in studies related with hydrocarbon degradation. As you can see in the table, there are specific strains that are mentioned in some species, and most of the species were isolated from soil that has been contaminated by oil. For your information, all of these Pseudomonas species are from different studies, and most of the result states the occurrence of bioremediation of hydrocarbon contaminated soil. Moving on to the explanation for mechanism. So firstly, the microorganism will produce specific biosurfactant to emulsify the pollutants or hydrocarbons, which then the emulsified hydrocarbon will be absorbed by the microorganism through endocytosis or active transport and passive transport. Then, the adsorbed microorganism will undergo an enzymatic reaction with the corresponding enzyme, which eventually enables the hydrocarbon degradation. This figure displays the mechanism of hydrocarbon degradation, but specifically for rhamnolipid. Moving on to discussion. So from my article review, the 5 Pseudomonas species that has been found to aid in bioremediation of hydrocarbon contaminated soil include Pseudomonas aeruginosa, Pseudomonas fluorescens, Pseudomonas putida, Pseudomonas stutzeri, and Pseudomonas lundensis. And among these 5 species, the most commonly used by study are Pseudomonas aeruginosa, which is believed due to availability of many various strains that has been identified by some studies, as well as having better degradation compared to other species, such as in a study by Ramadas et al. Stated that the percentage of total petroleum hydrocarbon degradation are more in Pseudomonas aeruginosa TPHK4 compared to the Pseudomonas putida TPHK1. And as for the mechanism of Pseudomonas species, they also produce specific biosurfactant, which is known as rhamnolipid, to degrade hydrocarbon. Next is the suggestion and recommendation. So most of the articles found in my review are international originated and can be analysed that there are lack of local research regarding this topic. Therefore, this review suggests that local research should improve and investigate more about the involvement of Pseudomonas species in degradation of hydrocarbon in soil, thus to propose more potential microorganisms to the local bioremediation industry. As can be seen in table 2, soil contamination are also potential to occur in Malaysia. Next is my list of references. And that is all from my presentation. Thank you.
Okay, so I open for Q&A.
Yeah, thank you very much for your interesting presentation, Ifa. So Ifa, I have some question. Can you go to your the coming slide? Just scroll down a bit.
Okay, jap. Lagi.
I think after introduction. After introduction.
Slide delete sikit kan?
Quite like. Sorry, jap.
Wait. A little bit down. Okay, here. Right, thank you. The respiratory tract infection, blah blah blah.
Wait a minute, for a bit.
Tak apa.
Still lagging. This one?
No, the one before. Atas lagi kot.
Kita nampak lain dia dah cerita lain.
This one, alright. So, according to your slide, you have already stated that the effects to humans such as respiratory tract infection, skin, kidney, blah, blah, blah. So, I'm just imagining, if you said that it caused all this type of bad effects, does that mean that all these hydrocarbon in the soil are volatile enough that even without doing anything, we can actually be exposed to all this? Bad effects of hydrocarbon contaminated soil, or if you use the soil for agriculture, then it will cause that such effect. You understand my question?
Yes, I do understand your question. Thank you. I don't do further study about that, but in my opinion, it must be how can we say it? We must enter through our mouth. For example, this study meant to say that if the human ate the contaminated plant.
Okay, meaning that you're doing agriculture on it. Okay.
Yes.
All right. Okay, that's the first question. And then you have put some contributing factors that helps in getting a good bioremediation process, for instance. So you put pH and temperature. So does that mean that you're talking about in situ remediation?
So far that I've read, many use in situ bioremediation. Yes.
Meaning that they are spraying the Pseudomonas or any types of bacteria to the contaminated soil, and it will voila, that happens. For instance, they akan terus it will
Yes.
remediate.
All
Alright, last question. I've
Okay.
understood that you said that rhamnolipid is some kind of biosurfactant, meaning that
Yes.
it is, what did they do actually?
It actually helps to emulsify the pollutants.
Okay, emulsify it, and then it's still
Uh-huh.
not degraded, right? So what is the next coming step?
As you can see in this figure, it will emulsify and form micelles of the hydrocarbon so that it can be absorbed by the microorganism.
So the microorganism will actually degrade all those contaminants by using enzymatic reaction.
Yes.
So what are the enzymes usually used?
From my there's not much studies that I found regarding enzyme, but one of the study that I found is they found the microorganism has alkylase hydroxylase enzyme something to which can hydrolyze the the hydrocarbon.
Okay, okay. Thank you very much for the question. Time is also up, Dr. Wan.
Okay. Alright, thank you, Pomfit. I think that thank you, Ifa. Okay. So
I can stop sharing.
yes, start sharing. So we move to the first presenter, no Shafika. So are we going to the other student? How about can we ask them to, or do they have to stay for the other student? How about the panels? Any suggestion?
Because every student's questions was listened by the rest. I don't know.
So we have to okay. Okay. So
But who's going to present?
okay lah. From the YouTube kan? Siapa
Okay.
boleh present? Fit boleh? Saya tak try copy. Tak boleh lah
Saya
nak
tak
copy.
buka lagi. Sekejap, I'll try.
Hold on
Okay.
I try copy. Tak boleh nak copy tadi.
Maaf lah sebab apa problem ni, sorry, maaf semua.
Tak apa. Kita try tolong mana yang boleh ya, Syafika. Fala boleh tolong tak, Fala? Macam mana nak buat ni, Fala? I try copy dia punya YouTube.
Saya present ni boleh tak? Tengok, boleh tak?
Tengok
Sekejap
Yes. Saya tak ada telegram dalam
ya ke?
laptop.
Okay. Tak apa. Pomfit dah cuba ni. Kita tengok macam mana.
Actually kan dekat sini dia tulis untuk present best for video and animation by tab, tapi bila saya buka, sebab saya dah buka dah tapi tak nampak kat situ. Sekejap saya try lain. Saya share ni. Tengok boleh tak?
Dia kalau video mengetat saya tak.
Nampak tak?
Sekejap. Dia presenting. Nampak. Okay.
Okay, saya on. Saya tak tahu kuat ke tidak ni.
Okay.
Aku beritahu No apa?
Suara dia tak project.
Sekejap
Sebab request boleh tak?
Saya try kuatkan. Hopefully okay. Sekejap saya try kuatkan semua means. Okay.
Assalamualaikum everyone. My name is Syarika Thomas.
Okay.
Over 2 years, we just made a study on clinical laboratory data for respiratory infection from KPG hospitals in 2015 and 2016. I will begin with the background of the study. Respiratory infection is a contagious infection caused by a variety of pathogens. It is caused by either single or multiple infection pathogens. Next is problem statement. Respiratory infection is one of the main causes of death globally. At least 2.28 million of deaths were recorded globally in 2016. The high statistic clearly highlights the importance of studying the respiratory infection pathogens. In Malaysia, there are limited epidemiological data available. Most data released more than 10 years ago, which is not relevant right now because virus keeps on evolving throughout the years. That's why we need an update on circulating respiratory pathogens to reduce the health burden of respiratory infection in Malaysia. In this study, there are 22 pathogens be detected by the multiplex real-time polymerase chain reaction. There are three objectives altogether in this study. First is to analyze the demographic characteristics such as age and gender of patients positive for respiratory pathogens detected by multiplex real-time PCR. Next is to determine the diversity of the circulatory respiratory pathogens by types of infection and the region of the KPG hospitals. Lastly, to identify the temporal distribution of the respiratory pathogens by monthly. We now move to the literature review. The first one is respiratory infection and common pathogens. Respiratory infection is a contagious disease caused by virus and bacterial infections in the respiratory tract. Next is the method of detecting the respiratory pathogens, which is real-time polymerase chain reaction. Then the common viral pathogen is rhinovirus or enterovirus, influenza A, metapneumovirus, and respiratory syncytial virus. For bacterial pathogen, the common one is Macroplasma pneumoniae. Last one is about KPG hospitals. There is a reason why we use clinical laboratory data from this hospital. This is because the KPG hospitals are one of the best private healthcare with 26 branches distributed throughout the nation. So we can say that the data from KPG hospitals could be used to see Malaysia as whole. Next is methodology. So basically, this study is a retrospective one, which is approved by KPG and UATM Ethics Committee. The social demographic data and clinical history of patients suspected with respiratory infection are archived. The DNA from clinical specimens of the patient are extracted in the laboratory and then subjected to the multiplex real-time PCR. Finally, the data obtained are analyzed using descriptive analysis and statistical analysis like chi-square test. For result discussion, we will first go through the demographic characteristics of the patients. The highest number of patients according to gender are male patients with 770 cases. Male have higher cases than females because their peripheral areas are smaller during their early years of life, which may expose them to respiratory tract infections. Based on age, the majority of the patients are infants, with 677 cases. Infants are more prone to respiratory infections due to underdeveloped immune system. Most of the cases, which are 631 cases, come from the northern region, which are Perak, Kedah, and Penang. Now we move to the laboratory results of studied population. From a total of 1,400 suspected respiratory infections, only 881 are positive. There are 655 patients of single infection cases and 226 confirmed cases for multiple infections. When sorted by gender, male patients have more cases for both single and multiple infection cases, which is 363 and 122 cases respectively. The same goes when sorted according to the age. Infants are the most patient for single and multiple cases, with 327 and 131 cases accordingly. Now we go further into the single respiratory infection. The most prevalent virus is rhinovirus or enterovirus with 176 cases, while bacterial pathogens is Mycoplasma pneumoniae with 11 cases. As usual, the most number of cases by gender for both pathogens is male. In terms of age, infants are majority infected by rhinovirus or enterovirus, while for Mycoplasma pneumoniae, the most affected are children. For multiple respiratory infections, it is divided into two, which are dual pathogens and multiple pathogens. The predominant pathogens reported for dual infection cases are influenza A and influenza AH3 with 25 cases. For multiple infection cases, the predominant pathogens are influenza A, influenza AH3, and rotavirus with 3 cases. As you can see in the table, for gender, females are the majority patients that infected with multiple infection cases. This is because females have smaller ostia of the paranasal sinuses and therefore more susceptible to sinusitis. The last one is distribution of respiratory infections and number of patients by month. Based on the graph, we can see that the peak is on September, and the most detected respiratory pathogens is rhinovirus or enterovirus with 35 cases on that month. The trend of the number of cases are related to seasonality. The pathogens tend to be active during autumn and winter, which are from September to February. This correlates with the month peaks of the pathogens in the graph. In conclusion, the demographic characteristics of patients really affect the rate of patients infected by respiratory pathogens. Male infants are the most susceptible for respiratory infection due to poor immune system. To answer the second objective, the diversity of the security respiratory pathogens by the types of infection are mostly single infection cases. The predominant pathogen for single infection is rhinovirus or enterovirus, while multiple infection cases are influenza A, influenza AH3, and rotavirus. Last but not least, the rate of respiratory infections by the pathogens are according to seasonality. The peaks begins from September to February. It is recommended that the reported prevalent circulating pathogens for respiratory infection should be utilized as a basement for any treatment plan. That's all from me. Thank you.
Thank you, Kongsi. Syafika, you boleh dengar ke? Boleh cakap ke Syafika? Syafika? Dia boleh cakap tak Syafika?
Tengok tak mute tu, tengok tak mute.
Saya boleh dengar tak? Dengar tak?
Boleh. Okay. So bijak soalan lah kan? Boleh dengar.
Alright.
Saya ada satu soalan.
Siapa nak tanya?
Ada, tanya. Boleh, silakan.
Boleh dengar tak?
Boleh.
Hello?
Boleh Fala. Boleh Syafika.
Testing.
Hello, Syafika.
Boleh. Dia dengar tak Syafika?
Lambat.
Okay, Syafika, your
Okay.
sample is from KPJ Hospital Malaysia. Syafika?
Dia lagging sikit kot.
Syafika? Dia lagging je. Tak apa.
Ya, saya dengar
Saya rasa dia dengar tu.
Okay.
Dia teruskan soalan.
How can it be?
Tak apa.
How can it be related with the seasonally autumn and winter?
I'm about to ask the same question just now.
Okay. Can you share the whole question, Syafika?
Actually, the seasonally peak, the autumn and winter. Actually, I found. Yes, I can hear it. Okay, I will answer it. Actually, the seasonality for autumn and winter, I found it when I read the literature review. I found that the months the same like the graphic that I got from the KBG data. So that's why I use that as justification. Something like that. And it's from autumn to winter. So it's September to February, which is actually correlated with the graph that I plotted. Is it answering the question?
Okay, Fala, did she answer question? Any question to Syafiqah?
Okay.
Okay, Syafiq ada soalan?
I have a question.
Okay.
Because you talk about infection just now, you said that it's higher in infants, and then according to what you have shown, the ones for patients more than 65 years old, the number of respiratory infection is not that high. So may I ask, because from my knowledge, it seems that the older people are more prone to get infection, especially by respiratory tract. Okay, so why is that it is low? Because as compared to young adults or adults, they are more, more resistant to infection. But why are the older people more than 65 are also not shown to have high respiratory tract infections in your study?
Okay. Okay. I think it's because for infants they have underdeveloped immune system, but for the elders they already developed their immune system but only weakened. So like if the respiratory infection invade the elders, the immune system can fight back because they have the memory, like they already live for a long time, the elders, so like they may have developed some mechanism to like macam mana nak cakap? To not get infected by the respiratory pathogen, and maybe they also have already know by experience, so they can avoid to get respiratory infection by doing something like wear mask to go out or something. But for infants, I think they don't have any experience for that because they only born they only born, so like there are no experience for them yet to get to get infected by the respiratory pathogen, so they easily get infected by it. Is it answering the question?
Okay, actually the infants, their parents will do it for them, okay, to avoid. But I think one of the factors, one of the contributing factors is because most old people do not go to KPJs. Because when you're old, you are more like, get more, you have many problems. So usually they will refer to government hospital. Do you think it's true, Dr. Wan?
Maybe, I think so. Okay. Yeah, because this KPJ is private kan. Okay. So
Okay, that's all.
yeah. So I think that's all. Okay, thank you Syafiqah. Okay. Syafiqah, thank you Pak Andy, thank you
Okay,
Mungkin, okay, for
thank you
helping
everyone.
Syafiqah, okay, for her presentation. Alright, so I think that's end of our presentation for FIP today. So I would like to thank the examiners, Dr. Fala, Pak Erni, Pak Fida, okay, and also to all the students, congratulations, you have done your presentation parts. Okay, so the next, I think you can refer to your coordinator. What is the next step? Okay, so I think with that,